The chemistry of life. Chemistry is what dictates life around us, so let's dive into it. We'll look at the building blocks of molecules, the amazing substance that is water, and the biological macromolecules you know and love, even if you don't realize it yet. When we talk about the physical world, we're talking about matter. Matter is anything that occupies physical space and has mass. Matter is organized as recognizable elements with specific chemical attributes. There are 118 known elements, but only 92 of them exist naturally. Of those 92, 30 are regularly found in living cells. Let's talk a little more general before we get super specific. Every element is an atom. The basic structure of an atom is essentially a sphere with a dense core and a cloud of moving electrons. Here's a schematic of that. In the center, you see two particles known as protons and neutrons. Neutrons carry a single positive electrical charge and act as the identifying hallmark of an atom. Your number of protons indicates your identity. The other nuclear particle is a neutron, which is neutral in charge and acts simply as a structural element. Surrounding the nucleus is an ever-growing cloud of electron shells. They carry tiny negatively charged particles known as electrons. In the normal neutral state of an atom, the number of electrons is equal to the number of protons, which balances out their overall charge. An atom is neither positive nor negative in its natural state. Arranged in columns and rows based on the characteristics of the individual elements, the periodic table provides key information about all of our elements that we currently know and how they might interact with one another to form different types of molecules. Most periodic tables provide a key or a legend of information to show you what they contain. You can see one on the bottom left-hand side that looks at hydrogen. There's a shorthand chemical symbol, a full name, an atomic number, which is simply the number of protons, and the atomic mass, or the total weight of the element in Earth's gravity. This is important to remember because we identify elements by their atomic number, which again is the total number of protons. This number does not change under normal circumstances. The mass number is the addition of protons plus neutrons. So if you need to figure out how many neutrons an element has, just subtract the atomic number from the mass number. You might notice that in this instance, both of these are carbons, but the mass numbers are different. That's because elements can fall apart by losing those structural neutrons. When that happens, the elements are known as isotopes of one another. Isotopes form at very predictable rates, and we can use their rate of creation to date biological samples that are centuries year old. Chemistry has a practical use. Who knew? One of the most important aspects of the study of chemistry is the focus on chemical bonding. All elements have electron shells where the electrons exist in constant motion. 2 electrons fit in the first shell, 8 fit in the second shell, and another 8 fit in the third. Atoms are more chemically and structurally stable when the electron shells are full. They can share, give, or take electrons to reach that state, and they'll do it in whichever way gets them to a full shell the fastest. This movement is chemical bonding and can be defined as chemical interactions. We'll talk about 4 different types. Elements tend to fill their outermost shells with electrons. To do this, they can either donate or accept electrons from other elements. When they do that, they're called ions, as they have a different charge. Cations are positive and anions are negative. These ions, as seen here, have varying charges. One is slightly positive, one is slightly negative. Due to this variation in electrical charge, the ions stick together in a chemical bond known as an ionic bond. This is one of the most famous ionic bonds. Sodium alone has 1 electron in its outer shell and is both dangerous and poisonous. Chlorine alone is missing 1 electron from its outer shell and is a very dangerous gas. Chlorine gas is how we won World War I. But when you mix them together and they form an ionic bond, they make table salt, which is something you need to have in your body. While ionic bonds are strong, they aren't the strongest potential bonds that can be formed. That honor belongs to covalent bonds. In covalent bonds, rather than electrons jumping from one atom to the other, the electrons are shared with other molecules. Two molecules missing single electrons, they can share one electron, and we call that a single bond. Share two, that's a double bond. When one atom draws the electrons nearer to itself because of a size difference, that bond is called a polar bond. We say polar because the resultant molecules have slightly positive and slightly negative poles because of the unequal distribution of electrons when compared to the centralized protons that won't move from the middle of the nucleus. In some covalent bonds, electrons are shared equally by all members, and those are referred to as nonpolar bonds. They tend to be less chemically reactive than those that are polar in nature. Polar bonds cause interactions known as hydrogen bonds. A water molecule, seen here in triplicate, is a polar molecule. The large oxygen draws the shared negative electrons nearer to it, causing it to hold a slightly negative charge. The hydrogen is therefore slightly positive. The positive hydrogen of one molecule is drawn to the negative oxygen of another. This slight interaction is known as a hydrogen bond. They're the weakest of the three chemical bonds we've studied by far. The fourth type of bond is actually just an interaction known as a van der Waals interaction. This occurs when electrons, which are always moving, clump together by chance on one side of a molecule. It sounds a little strange, I know. Here's what it looks like. I always imagine electrons as being equally distributed because that's how we draw them, but that's not the case in the natural world, as you can see here. As electrons are constantly moving, these interactions don't last very long and they're extremely weak, but they can affect the chemical properties of some solutions. We're going to focus on hydrogen bonds though for a while because they have a much bigger impact in the world around us. Water has many interesting qualities thanks to the properties brought about by hydrogen bonding and its polar covalent bonding pattern. Here we see a macroscopic image of oil and water. Oil is a nonpolar compound and hence will not dissolve in water because it can't mix with the polar molecule. The hydrogen bonding between water molecules causes the lattice structure of ice. The bonds form and freeze in place, holding the water molecules a little farther apart from one another than they usually lie. This locked structure makes ice less dense than freely flowing molecules of liquid water. Ice's lower density enables it to float. The polarity of water also allows it to be an excellent solvent. A solvent is a substance that can cause something else to dissolve. Here you see how water causes table salt, NaCl, to dissolve. The positive hydrogens surround the negative chlorine anions, causing them to release positive sodium. The negative oxygens surround the sodium and do the same. When something seems to disappear in water, it's just falling apart and you can't see the individual molecules anymore. A final property of water that you definitely can see is surface tension. Hydrogen bonding happens on the outside of a body of water, no matter how small, and it causes tension across its surface, as seen here. This allows water to stick to itself, which is called cohesion, and it allows it to stick to other things. That property is called adhesion. You've seen it if you've ever used a straw or looked out a rain-covered window. Water is capable of dissociating or falling apart into its component parts and becoming a single hydrogen, that's an H+, or a hydroxide ion, that's a negative OH. Both of these component parts turn out to be highly reactive and potentially dangerous on their own when in high concentrations. We chart their concentrations on a pH scale that ranges from 0 to 14. At 0, a solution is pure H+ ions and is considered acidic. At 14, a solution is pure OH and is considered a base, or you can say it's alkaline in nature. At 7, only H2O exists and the solution is neutral. Now, it's important to remember that not all H's and OH's actually come from the dissociation of water, but that's where some of them can. There are certain substances that naturally donate H+ ions and others that naturally donate OH ions. It's just easiest to talk about the ones that come from water. pH balance is an incredibly important part of homeostasis, or our internal cellular controls. Your body's pH is an average of 7.45, and it's dangerous to push your body out of that range. Although your kidneys and lungs do an excellent job of maintaining your pH on their own, you can get ill when your pH ranges due to great illness. Some people have started selling products that are meant to alter your natural pH balance of your body. While most of them are useless, some can actually be quite dangerous. Water is the best thing you can drink, and I mean just regular plain old water. After years of studying biology, there's not a lot of promises I'm willing to make, but that one I stand behind. Don't drink weird water, drink regular water. It's the best thing for you. Let's move on to chemistry that is more biological in nature. We can talk about biochemistry, which focuses on life that is carbon-based. Carbon has 6 protons and therefore 6 electrons. 2 electrons in the first shell and 4 in the second. Carbon, with its 4 empty spots in that outer shell, can therefore form up to 4 chemical bonds. Life on our planet is all carbon-based because of this. It's the backbone of all biological macromolecules that we currently know of. When I say backbone, I mean that it's literally the central physical element in all molecules. Here you can see a few different examples. First, you see stearic acid, which is a component in all animal fats that we use to make hormones. It has a long chain of carbons down the center. Glycine is the second molecule, and it's a central component of many proteins. Glucose, the molecule on the bottom, powers all of your cells, and it has a 5-part carbon ring as its backbone. We look for carbon on other planets because all we know is life that is carbon-based. We'll focus now on biological macromolecules, the first being carbohydrates. Carbohydrates are made out of long chains of individual molecules of ring sugars called monosaccharides. The most common is glucose, which is cellular energy in animals. When you link 2 of them together, we call it a disaccharide. Di means 2. The most common of those is sucrose, which is the preferred energy source for plants. Polysaccharides, or many sugars, are studied in 4 large categories. Starches are storage polys in plants. Glycogen is the storage form in humans. We keep it in our liver and muscle cells. Cellulose is a polysaccharide that is a structural component in most plants. And finally, there's chitin. Chitin is the structural component of the exoskeleton of insects. Ever crunched a bug or cracked open a lobster? That's the chitin breaking that you're hearing. In case you were curious, all of those sugars are attached to one another by dehydration reactions. All of those units have hydrogens and hydroxide ions hanging off of their sides, which are capable of interacting with one another and forming a water molecule. The open bonding spots allows the individual pieces to bond with one another once the water is gone. We'll see this bonding pattern again and again in both proteins and nucleic acids. Just in case you're a visual learner, here's what some of those monosaccharides look like that we'll be studying. These 3 are all isomers of one another. That means they have the same structural components, they're just put together differently. Note that the hydrogens and hydroxides are plenty, and they could all participate in dehydration reactions to link these together in order to form different types of disaccharides. Polysaccharides are more complex in their physical structure. Here you see different linkage and branching patterns that are types of polys that I mentioned before. The more highly branched, the more strength a macromolecule has. The second macro for consideration are the lipids. They are energy storage molecules that are hydrophobic. They have a glycerol head with 3 carbons and 2 or more fatty acid chains attached to those. Those fatty acid chains, you'll see them in just a second, can be saturated or unsaturated. Saturated chains have all of the hydrogens that they can hold. Unsaturated chains are missing a few because some of the carbons form double bonds with each other rather than holding on to more hydrogens. Not so typical lipids include the steroids. They have that hydrophobic classification, but they exist as rings rather than tailed molecules. They exist in animals as multitasking molecules that commonly signal different physiological changes. The plant version of those are the waxes, which are used for protection and a safeguard against water loss. There are some instances of lipids protecting animals too, as in the instance of Senior Adorable Otterface here. His lipid coating gives him protection and insulation against the cold river water that he lives in. Again, for us visual folk, here are the molecules we talked about. The saturated fatty acids are straight molecules with every possible hydrogen bond they can make. The unsaturated acids trade hydrogen for a double bond that causes a kink in their tail structure. Up top, you can see the typical triglyceride. They have the 3-carbon glycerol head and 3 tails. Here's a cylindrical steroid molecule, and possibly the most important molecule in this image is the phospholipid. Every cellular plasma membrane that we know of is made of phospholipids. Their name is derived from the phosphate that's attached to the 3rd carbon on their glycerol head. One tail is saturated and the other isn't. We'll come back to this molecule time and time again when we talk about cells. I like to bring up trans fats in class because they're so dangerous to human health, although we still don't really understand why. A trans fat is a fat that has had its physical chemical structure changed. By altering the location of a hydrogen, the molecule can be forced to lay flat. These are 2D drawings, but remember, these are 3D molecules. For whatever reason, this allows the food item that contains a trans fat to last longer on a grocery store shelf. Foods with trans fats don't spoil as fast, but for whatever reason, they definitely spoil in humans. Eating trans fats causes heart disease and leads to heart attack and stroke. They've been removed from a lot of foods and have even been banned from some entire countries. Let's move from something you definitely shouldn't be eating to something you definitely should, and that's protein. Protein has many functions, all of which are tied to their structure. The structure of protein can be studied on 4 levels, labeled 1 through 4 in Latin. Those are the words you see here. The overall structure is so important to the function. They are complex and held together by numerous bonds and interaction. Ruin the structure, ruin the function. In order to deactivate a protein, you have to add copious amounts of heat, change the pH, or wash proteins in particular chemical substances. When you break down a protein, you end up with a collection of amino acids. They are the building blocks of proteins, and there are 20 different options. Amino acids have a central carbon, a carboxyl group, an amino group, a hydrogen, and an identifying R group. Here's some schematics of 5 of them. Amino acids, again, are made up of a central carbon bonded to an NH2, a carboxyl group, COOH, and a hydrogen atom. The central atom's 4th bond varies among different amino acids. Seen here are alanine, valine, lysine, and aspartic acid. The R group gives the amino acid its identifying chemical properties. It might make it an acid or a base, or maybe polar or nonpolar. Link a bunch of amino acids together and you have a polypeptide. Read off the name of the individual amino acids and you're simply reading its primary structure. Allow the primary structure to form its natural hydrogen bonds and you'll end up with alpha helices, that's seen here, it's that curly shape, or beta pleated sheets, which are crimped like accordions or Japanese fans. Proteins will fold those structures into 3D units that are cubed or spherical in shape. That's the tertiary structure. For most proteins, that's as fancy and complicated as they get. Some, however, work together like transformers. A few different structural proteins working together make a large functional unit capable of even more complex processes, and those are referred to as proteins with quaternary structure. Our final macromolecule is arguably the most important. Nucleic acids are long polymers of nucleotides. Nucleotides consist of one of two types of sugar, either deoxy for DNA or ribose for RNA, a phosphate group, and finally a unit called a nitrogenous base. The base gives the nucleotide its identity, just like the R group does in a protein. There are two types of nucleic acids, RNA and DNA. DNA is a double-stranded alpha helix with one of the four bases known as adenine, thymine, guanine, and cytosine. RNA, on the other hand, is single-stranded and has adenine, uracil, guanine, or cytosine. These two molecules will be our basis for our study of genetics, and we will talk about them a lot in later chapters. With that, we end chapter 2. So what's next? Read the book and take plenty of notes in your own words. Study in whichever manner works best for you, and take a crack at your homework.